Good afternoon. We're here in Barcelona, Spain for ESMO 2024, and I'm joined by Dr. Matthew Galsky from Mount Sinai, Tisch Cancer Institute, who earlier today presented on preliminary safety and efficacy of Disitamab Vedotin and Pembrolizumab in untreated metastatic urothelial carcinoma. Welcome, Dr. Galsky. Thank you for joining us.
Of course.
I would love to hear about the study that you presented today. Could you give us a brief overview and then discuss the findings?
So the study combined Disitamab Vedotin and Pembrolizumab. Disitamab Vedotin is an antibody-drug conjugate directed against HER2, linked via cleavable linker to an MMAE payload. It's been studied before, and it's actually studied pretty extensively in China, and they've already done a phase 1b/2 study of Disitamab Vedotin plus toripalimab, a PD-1 inhibitor in China. Very promising activity there. The drug's now being developed internationally, and so this is the beginning of the experience with the drug in an international population. So a small safety lead-in cohort of 20 patients treated with Disitamab Vedotin plus Pembro with a primary objective of determining the objective response rate per blinded independent central review.
And the patients that were included, I noticed there was HER2 low included as well, which is very interesting. Can you tell us a little bit about the findings in both groups?
Sure, so let me comment on the HER2 status.
Yes.
So the HER2 categorization is referred to in this study as HER2 positive and HER2 low. It's sort of arbitrary names, but that's just used to categorize the HER2 expression into two groups. It's important to remember that HER2 low refers to HER2 1 plus or HER2 2 plus negative FISH. So as this being an antibody-drug conjugate, it's unclear how much HER2 expression you need in a tumor. Of course, you need some probably in the data in other tumor types showing activity in patients with HER2 zero expression. Might be related to intratumoral heterogeneity and the way biopsies are done and things like that. But you probably need some, but how much you need, who knows for sure. So that's the categorization here. Even though we call it low, that could be 2 plus expression. And then patients with HER2 high tumors are 3 plus expression by aUC or positive FISH. 20 patients enrolled, safety lead-in cohort. Disitamab Vedotin is administered every 2 weeks. Pembrolizumab, the Q6 week dosing, so the cycles are every 6 weeks. And the objective response rate was 75% with a complete response rate of 35%.
Very good. And that's an important categorization you bring up in terms of HER2, because as you know that in HER2 tumor agnostic approval right now, the FDA approval is for HER2 positive, which is defined as 3 plus.
That's correct.
So that's why it's very interesting findings here with the HER2 low. So did you see any differences in overall response rates when you looked at HER2 positive versus HER2 low?
So very small experience so far, and while there didn't seem to be a major difference, it's important to remember two things. One, that the 70% of patients had HER2 low tumors, so our experience with the HER2 positive group is more limited. The response rates were 68%, 66% in the HER2 positive group and 78% in the HER2 low group. And so based on these small numbers, there does not seem to be a major difference, but there's a caveat there, and it's the caveat that you brought up before. So HER2 positive here is defined as 2 plus positive FISH or 3 plus. There's another tumor agnostic approval in 3 plus based on IHC. Whether or not there is some correlation between IHC expression and activity of this drug, we don't know that for sure. Earlier studies with single agent DV suggest maybe there is some, but of course we'll need larger sample sizes.
Yep. And in terms of safety, what did you find?
So safety profile of this drug is a bit different than Enfortumab vedotin. There's some overlapping toxicities, some that are non-overlapping. So we see fatigue, we see alopecia, we see nausea, we see neuropathy. The neuropathy so far hasn't been as well characterized, whether or not it's different, whether or not it happens at the same time. We need more data there. We see some rash, but we don't see high-grade rash, and so whether or not the rash is being driven by DV or mostly driven by Pembro in this combination, I think we also need more data for that. And we don't see any ILD in this cohort. And there were no grade 5 events in the small cohort. So I think we're seeing a safety profile that's similar but distinct from Enfortumab vedotin.
which is important because if we're going to see, MMAE-based ADCs moving into the perioperative space eventually, and we're talking about different sequencing, toxicity profiles are going to be very important in terms of overlap. So, what are the next steps for this study in terms of phase 3?
So there are several additional steps. One additional step is that there are other cohorts in this study that are currently enrolling, and those include single-agent DV in patients who have progressed despite prior therapy but haven't had EV yet. And so that's single-agent DV, that's sort of the immediate term development. For the combination, there are two ongoing studies. One is this study in part 2. In part 2 randomizes patients between DV Pembro versus DV alone to assess contribution of components, still with a primary response rate endpoint. And then there's a phase 3 that's randomizing patients between DV Pembro and platinum-based chemotherapy enrolling internationally.
Very good. So very exciting time to have new targets for, and especially as we discussed, with the perioperative landscape rapidly evolving, and having an additional target with HER2-directed therapy may be helpful, especially if we do end up seeing a signal in HER2 low patients. So very exciting time. Are there any other findings that surprised you?
Not necessarily that surprised us, although it is very nice to see the response rate aligns really nicely with the experience in the study done in China. That study is being updated at ESMO as well, and overall response rate of 75%, exactly the same as we saw. So that's nice to see. And I think all of this data collectively really establishes HER2 as a bona fide target in urothelial cancer in the U.S. Of course, there's an FDA approval in a tumor agnostic study, and so I think that reinforces the need that we need to do this testing now as part of our standard treatment approach.
And perhaps where else do you see, with the rapidly evolving landscape, both in perioperative and the locally advanced setting, where do you see DV's role?
So I think DV has the potential to move up earlier, and I think there are two unknowns that some of them you alluded to before that will really dictate the development of this therapy. One is what the activity is of EV in a HER2 overexpressing population. We don't know that. We sort of assume that it's going to be similar, but we actually don't know that. And these tumors, I can say from anecdotal experience, there's something unique in terms of the natural history, in maybe even sites of metastatic spread. And so I think that these are different, and we don't fully understand what EV Pembro does in a HER2 overexpressing population. The second is what you alluded to before, which is that I think we do need to understand a little bit more about the HER2 biomarker before just saying we didn't see any difference between the two groups. Because if you look at the depth of response, there is actually, if you squint, there's sort of a correlation between the two in these small numbers. And so I think it's quite possible that there will be a subset of patients that we can identify that really should get a HER2 targeting approach rather than another approach.
Yeah, so in terms of sequencing, that'll be very important we'll learn that over the next few years. So.